a lot of people in the field that were very well respected said there's nothing you could do about this. And they said it's really hard to develop drugs for disease, especially genetic disease. And I said, watch us. Drug development is really, really hard. But you know what? Living with rare disease is so much harder. So no one's going to tell me what hard is until they live a single day in my life. There was this valley of death that we were sitting in which early research and discovery scientists, neuroscientists, and people that were really interested in understanding the genetics of this disease have done a ton of work to understand the biology, create the models, understand what drugs could and couldn't work. But they were not drug developers. And that's the valley of death. Where does neuroscience meet clinical medicine?
Hi, I'm Ron Kleiman, and this is Generation Hope. I'm deeply honored to be speaking with Dr. Allyson Berent, a veterinarian, interventional specialist, researcher, biotech founder, rare disease mom, and chief science officer of FAST, the Foundation for Angelman Syndrome Therapeutics. Allyson's daughter, Quincy, was diagnosed with Angelman syndrome, and that diagnosis changed the direction of her life. What followed is an extraordinary story of science, advocacy, urgency, and a parent community that refused to wait. Through FAST, Allyson helped drive one of the most important therapeutic programs in Angelman syndrome, GTX-102. What began as a parent-led effort has grown into a fully enrolled global phase 3 study, with GTX-102 data expected in the second half of 2026. And that work has continued through AS2 Bio, a drug development accelerator helping advance the next generation of Angelman therapies, including approaches in gene replacement, stem cell-based therapy, and gene editing. We talk about how that happened, what it takes to move a treatment from an idea to a clinical trial, the realities of building therapies for rare diseases, and why families are often the force that pushes science forward. This is a conversation about hope, but also about the work behind hope, the science, the setbacks, the strategy, and the belief that these disorders can be treated. If conversations like this are important to you, please subscribe, like, and share. It's the best way to support Generation Hope. And now, Dr. Allyson Berent.
Good morning.
Good morning.
Thank you for this.
Yeah, welcome.
I really wanted to start with, what's your earliest memory of a purpose, of what you got into in terms of what you studied, veterinarian stuff? What shaped you to kind of go that route?
Going into veterinary medicine was actually, it's a really strange reason that I started wanting to become a vet. And it's not actually because of purpose and animal care and love and, ethics and morality at all. I was about 7 years old, and I was in the car with my father and my sister. And my sister's about 3 years older than me, and her best friend was in the car. And my dad asked her in the summer, we were on a ride going somewhere, what do you want to be when you grow up? And she said she wanted to be a veterinarian. And I thought she was so cool. She was so pretty. She was so sweet, very popular, just a nice girl that I really admired as an older girl, a role model. And she was my sister's friend, and I always wanted to play with them, and I always wanted to be around them. And I just really admired her for no great reason other than she wanted to be a veterinarian. And I was like, well, she wants to be one, I want to be one. And so it was the middle of the summer, and then we went to school the next year, and I was entering third grade. And my third grade teacher said, what do you want to be when you grow up, around everybody in the classroom? And I was like, a veterinarian. And she was like, well, it's really hard to be a veterinarian. There's only 26 vet schools in the country, and it's harder to be a veterinarian than a human physician. So you really need to get straight A's. And essentially she was saying, don't get your hopes up. It's really hard. You might be disappointed if that's really what you want to be, because it's really hard. And I was like, well, then I definitely want to be a veterinarian, because I was just given the challenge. And that was kind of my personality. It was like, if someone says you can't, then I would say I will. And that was very clear at that age that was my personality. And so that moment on, I only wanted to be a veterinarian. Nothing else mattered to me.
Wow.
So 8 years old, 9 years old, 10 years old, and I just waited until I could get my working permit. And when I was 14, you can get a working permit. I volunteered at a animal hospital for the first time. But my cousin, my father's cousin, so my second cousin, was a veterinarian. And my dad took me for the summer to spend some time with his cousin. And I got to watch just traditional general veterinary surgery and appointments and seeing how the animals are treated in the veterinary office. And it further solidified that I wanted to be a veterinarian. At this point, I'm maybe 10 or 11.
Wow.
And then at 14, I got my working permit. I went to the local animal hospital in our town and I asked if I could get a job. And they gave me a job on Saturdays to clean cages and to assist the veterinarian, and I did it. And then that progressed to getting, another job in a marine biology, doing a marine biology camp. And then that became interested in aquatic animals. And suddenly I was on the path to being a veterinarian. And I only wanted to go to one college because it was the best veterinary school in the country, Cornell University. So I wanted to go to undergrad at Cornell so I can get into Cornell's vet school, which I did. And so it just became this perpetual commitment I made at age 7 that I would do. And then I fell in love with veterinary medicine because I was in love with the idea of being a vet, not because I was in love with animals.
Wow.
So it's very interesting because most veterinarians are in love with animals and that's why they want to be a vet. I fell in love with animals eventually, but that wasn't why.
That's fascinating.
Yeah.
That's fascinating.
Yeah.
How is that attribute of your personality? A lot of
personality traits have a strength and a weakness.
Yeah.
That's fascinating.
How do you see?
That's fascinating.
How do you see that?
Yeah. So I think the weakness is that it did not leave me open-minded.
Wasn't why.
I was so
That's fascinating.
I had such tunnel vision to do
How
that.
do you see
That's fascinating.
If I step back now and think, what did I actually want to do
personality traits?
versus what did I commit to
why.
do?
That's
I probably
fascinating.
would have done something different.
How
to
do
be
you
honest.
see?
That's
And
fascinating.
I love medicine, so I don't think it was totally wrong. But I think I would have.
Wasn't why.
I would have probably gone into.
That's
human
fascinating.
medicine. Now, I am driven naturally by having impact. I'm driven by wanting whatever I do. I always want. I would have probably gone into have
That's
a
fascinating.
lasting impact in whatever it is. But my dad, so I don't think when I was really young, whatever room we
personality
walked improving the life of your child and the children of others like your child. I think that allows industry and investors and other people in the space that are not living in our shoes to realize, if they're humble enough to be willing to realize, that maybe when you cut off all the fat and you focus on what you have to accomplish, where timelines are not negotiable, but finances are limited, you actually need 1/10 the number of people with 1/2 the number of hours to actually get it done. But when you have layers of hierarchy and red tape and approvals, and this is their job and not your job, and his job and not her job, then suddenly everything takes 10 times the amount of time at 100 times the cost. And we don't have the luxury of that. And in some ways, it's almost our best asset because we don't take shortcuts. We do everything with rigor, but we realize, why do we have to do it that way if we can do it this way? And the answer is, let's ask if we can. We share it with the next generation of developers, which are generally parents. Because who comes to us for advice? Other parents, other people that want to develop drugs either for their kids or for their loved ones, or other startups, if they would open their eyes to the fact rare disease can't be treated like common disease. We don't have the resources. And so we realize what's possible. And we can be nimble and really deflect and pivot where we have to in order to accomplish the same goal, when we don't allow white space to exist on a Gantt chart. And when I look at big companies' Gantt charts, when I come in to give advice or whatever it is about Angelman or anything else, I always question, why is there so much white space? What is the dependencies here? What is on the critical path, and why can't you shorten that? And it's like, as soon as people see white space, they just fill it rather than pull it in.
Yeah.
You could fill that white space by pulling things in. You don't have to fill it by bloating it.
Yeah.
And I think that that's not always a common thought. I think really good developers think that way. I've learned that not because someone trained me. I learned that because I didn't know any better. And sometimes being naive and coming in not knowing any better, when I say, listen, I'm not willing to wait 6 more months for this. If we can pull this in by 6 months, why wouldn't we? What is it going to cost to do that? What is the risk and what is the benefit? And if the risk is a little bit of money, but we have to hire less people because we don't need as much time, that's not really a risk.
I'm going to jump ahead because of what we're talking about. We'll go back and forth.
It's fine.
But do you see a wholesale kind of change in direction in the industry because of what FAST has been able to accomplish?
I do. I hope so. I see it coming slowly but surely. We're approached by investors and industry every day. We need to raise money in order to be able to build this platform of a drug accelerator for rare neurodevelopmental disorders. That's the goal of AS2 Bio, is not to stop at Angelman syndrome. Take what we've learned in Angelman and apply it to every other disease in which it is applicable. Diseases that require knockdown, that require replacement, that require cross-correction. The things that are biologically similar to Angelman, most of which is neurodevelopmental disorders and single gene disorders, apply it for them so they don't have to do what we did in a silo. I believe very much in collaboration, and de-siloing is the only path for rising tides to lift all boats, and to not hold knowledge and learnings proprietary so that other people slow down. If I can allow another disease to do this in half the time we did, which should be the case, right? As a parent, you want your child to do better than you did. I want the next disease to do better than we did. That's the way you advance the field. And if we hold people back with what we know, then everyone will do the same thing we did in a silo, but maybe even less efficiently. So our goal is to not allow that to happen, because we share with everybody we encounter where we can, where it's applicable, they will move faster. Because of that mentality, there's impact investors that see that as, one, we can make a difference in the world, and two, you're going to make money because you're going to show success. And the faster you move with careful rigor, and I emphasize careful rigor, because you don't want to be, this is one of the problems with parents developing drugs. They want to move so fast, they don't understand the risk. And as a clinician and now developer, you have to balance both. You have to move fast with rigor and with safety in mind first and foremost. And parents doing this have to know that, and a lot of them don't. So, and unfortunately, what will happen is there'll be a devastating event. and it will set the fields back if people are not careful. So having that care and being rigorous, efficient, and thoughtful is really important. And when investors see we have those parts to the ecosystem we need for Angelman, they're like, I want in on that. Because if I give you more money and give you more resources, I know you're not going to waste them. You want a small, nimble team, but you want to grow just enough to accomplish your Gantt chart and get that done quickly. And the faster you eradicate Angelman syndrome, the faster you'll bring on these other diseases in the background. And if we can employ all of your learnings for other diseases, now you truly have an assembly line and a platform where this team understands the meaning of rigor and being efficient with time and money. And there you have low costs with high reward. And that is really being seen by investors now, I think.
See, seems like a no-brainer for an investor.
It seems like a no-brainer, but people also want to believe in people that have been successful.
Of course. Yeah.
And so they look at you and your ideas and they're like, yeah, right. And that's where we were 5 years ago, is good idea, yeah, right.
People are paying attention.
Yeah, look, you think you can do that. Guess what? It's really, really hard. And I always say, drug development is really, really hard. But you know what? Living with rare disease is so much harder. So no one's going to tell me what hard is until they live a single day in my life, as you do.
Yeah.
And that is hard. Drug development is nothing compared to that.
Yep.
And so if you have the ability to handle the life of a rare disease family, you have the ability to handle drug development. It's hard, but you figure it out. My kid has a seizure. That's hard.
That's hard.
I figure it out. We get through to the next day, and guess what? Next week's a better week. So we know what hard looks like. We know how to navigate hard, and we know that there can be better days on the other side of hard. So this up and down of drug development is like perfect for rare disease parents, because nobody understands it better and knows how to not give up than a rare disease parent. And a lot of industry partners, they give up. They find another disease that might be less risky, and something goes on the shelf in order to do a better bet. Guess what? We're not gonna be afraid of a hard bet. We're gonna know when to call it on a bad bet.
Yeah.
That's the difference. You can still push hard bets. You can't push bad bets. And you have to understand what's best for my kid. I'm not just gonna push something for the sake of pushing it if I don't think it's a good bet. I'm gonna push it if I think it's gonna make a difference in her life.
Yep.
And knowing that, people don't give us enough credit. They think that, well, parents are just gonna keep pushing even when they shouldn't. No, you need science to drive your decisions. Is this go or no-go decision has to be based on science. And if science says it's a no-go, it is a no-go.
Yeah.
Science needs to define that. But if it's science says it's a go, even though it's a hard bet, we're gonna go. Because we believe that we should follow the science, the money will follow.
Yeah.
We can't follow the money.
100%.
There's 2 of us that are Angelman parents. We have a team of 12 at AS2 Bio. The rest are not Angelman parents. They just believe in the mission. And they're gonna call out bad decisions. They're gonna call out scientific no-goes, just as we will.
Yeah.
But we're gonna find a way when the science says we should go. That's the key.
I see so much that loops back to that one thing that your dad told you.
Mm-hmm.
Tell me about your dad.
My dad's awesome. So my dad is a really interesting guy because his personality is like almost the dead opposite of mine. I'm actually much more like my mother. My dad is really laid back. My dad's not somebody who is going to, want to be the CEO of a company and the best at this or that, or make whatever he does into the greatest possible thing of all time and have that type of impact. My dad wants to have impact in the room he's in. My dad wants to make people laugh. He wants to make sure that he's a good human. He wants to make sure he raises really good, strong, I have a sister, so we have 2 girls. He wants to raise 2 strong, confident women. He wants to be there for us at all call. He wants to make sure when we have a hard day that we can call him and he'll always drop everything for us. And his goal is to just make you laugh and make sure that we're provided for. But he also wants to make sure that we're not spoiled, that we work really hard to make a life for ourselves, and that we are humble and know where we come from. And he is not the type A personality that I am at all. He's like the hopeful, kind, very green personality. And I'm the very red personality. And so we're very, very different. And I think that's why we get along so well, because he brings out like that green to my red.
Yeah.
And really the push-pull between us is very, very nice. I can go to him with a tear, and I can go to him with an accolade and an award. And he is there for me in the same light with pride, regardless of whether it's a hard conversation or an easy conversation. So he's just a really good guy.
And your mom?
And my mom is red. My mom is totally red. So I get that from her for sure. My mom is the most motivated person. She comes from very little financially. She comes from someone who her parents and her grandparents worked their butts off in Manhattan, New York, in order to get what they got to provide for her and her siblings. She's one of 4. She has a brother who has cerebral palsy. She has a sister who died of multiple sclerosis. And she has another sister who died of brain cancer.
Wow.
And so my mom's been through a lot in her life, and she was always the one who held it up. Her father passed away in, his, I think, early 70s. Her mother passed away in her very early 80s. So she really had to take care of her brother with cerebral palsy, who's still alive. And so she's had a hard life. And despite all of that, and despite them not having money or funds or resources to send her to college and to do all the things, she just really worked with grit. Like, she just figured it out, and she really navigated the world to make money for her family. So she was really the breadwinner in the family, and she was the one who had worked very long hours, very hard. She started her own company, her own business, from a spa and like a salon to owning her own title insurance company, and really, got into real estate because of somebody from her spa, and then real estate led into real estate law, and that led into title insurance, and then that led into starting her own title insurance business. And she really learned how to navigate a world of all men. Really, she was one of the few women in that world in the 80s. And she learned how to navigate that in a way in which she showed me that strength of, you need to provide for your family. And if you didn't start with something, you can still end with something.
Yeah.
So she gave me that, and she gave me the work ethic, which really my dad didn't have. My dad was more like, I'll come home for the kids, which is not wrong and not bad. But my mom's like, I'm going to be at work till 9. And my dad's like, I'll be home at 4.
Team effort.
It was a team effort, and they were very different. And so I knew early on that they were different. And I think in hindsight, I didn't realize it at the time, but in hindsight, I hope that I took the best from both of them. And not to say they're perfect, because none of us are, but they gave me and my sister a skill set, which is very different. My sister is very different, but a skill set to be autonomous. And I think that autonomy and that kindness together actually 100% is who I am. I like to work autonomously. I can think on my own. I know where to bring in help. I know where I don't need help. I like other autonomous people who have the drive to do what they need to do and do what they set out to do. I don't handle incompetence well. I don't handle lazy well. I don't handle people that just want to get by on other people's hard work well. So that intolerance of incompetence. That's my mother. And my husband, he's totally intolerant of incompetence.
I thought you were going to say something else for a second.
No, he's totally incompetent. He's the most competent person I know, actually. When you talk to him, he's like actually the smartest and most competent person I know. And so my mother teaching me that and then living to his standard has allowed, I think, a lot of the success of our veterinary, he's a vet too, so our veterinary career, our home life, and I think my biotech career, because he has taught me never take no for an answer. And you don't call it done until you've done the best job you can at it. Don't just accept mediocrity. Accept as close to perfection as you can get without wasting time. So it's that balance of get it right, don't waste time, don't waste money.
Yeah.
And so that, I think, is a combination of him and my parents for sure.
And your sister, so you guys are very different?
So different. So different. She's blonde, firstly. So different. She looks like my dad. I look like my mom. I'm a spitting image of my mother. My dad's blonde and fair. My mother is dark-haired and dark-skinned. Yeah. So I'm a combination. She's my dad. I'm my mom by the looks. But my sister is just a very calm, very sweet, heart of gold. She's a school teacher and became a principal and became a superintendent of schools. But she just like is a kid at heart in everything she does. She loves children. She will get on the floor and get dirty with kids. She wants to see the success of children and her teachers and her schools. Like her love for like the humanities, you know, is different than my love for the science. Like I am black and white and like pushing everything for an outcome. My sister is, kids are our future and we need to prepare them to be our future. And she has patience as a saint. I have zero patience at all. She is everything I'm not, in terms of like, she like is the yin to my yang of negativity. Like where I'm negative, she's positive in every way. She is like such a sweetheart.
Your family seems to be a perfect balance.
Oh, it's a balance. And it's funny because when we're all together, it's like me and my mom are the same and her and my dad are quite similar. But in the same regard, me and my dad get along incredibly well, and her and my mom get along incredibly well. And I think it's like the opposites attract in some ways. And my mom and I butt heads. I love her deeply and I thank her for everything that I am. But like, you know, we have different politics, we have different views on life, we have different, you know, expectations of people, and we call each other out on it because we're so similar. And my dad is just like, everybody leave it alone. You know, like my dad is the diplomat for sure. And so is my sister.
Wow. That's, we should all be thankful for that.
Yeah.
For everything that pointed you to where you are and everything that's happened. You know, it's that whole cause and effect kind of idea. And I'm just really thankful, and I'm sure most people are, that your sister's friend didn't say that she wanted to be a pilot.
Maybe or an astronaut. But literally, like I may have been that had she said that. It is really funny that is like where it started. And I do think my personality just like drove to that. It's very interesting.
Yeah, you know, it's follow your calling wherever it may go.
Wherever it may be, even if she's just a 13-year-old adorable little girl.
So let's shift to Quincy. So, your first daughter, Kai, was a couple of years old, I guess?
Yeah, she was 2 when Quincy was born.
2, okay.
Yeah, so she's 2 years older than Quincy. They're all about 22 months apart.
Okay.
So she was about 2 when Quincy was born. She was 22 months. And Kai was very neurotypical. She was like the quintessential perfect pregnancy, birth, toddler, everything. She, you know, had her first word at 7 months old, like ahead of the curve on every level.
Yeah.
She was the kid that you're like, you know, when I get pregnant, I want my first child to be a girl. That was her. Like I wanted a girl, I had a girl. I wanted a child in the summer, so my maternity leave was during good weather. She was born June 2nd. Like she was like everything that we planned. Yeah. She is a perfect mix of my husband and I. She's incredibly stubborn. She's now a teenager, so that is challenging, to say the least. But she is just a brilliant, beautiful, humble child. And she was everything we wanted. That first kid, she was that.
So she was roughly 2 years old, and Quincy came in. And how did Kai, as an older sister, react to a brand new baby? Yeah,
so Kai was very excited about the baby. She was super excited to be a big sister. Quincy was very different than Kai, and we knew that in day 3. Like Quincy was born, she was perfect pregnancy, totally fine when she was born. Everything was great. Yeah. Discharging from the hospital, she started refluxing, and Kai never had any reflux at all. And they're like, oh, happy spitter baby's reflux, no big deal. So we took her home, and it just continued, and she was suckling fine, and then it just got worse and worse to the point that she couldn't suckle. And so I was up all night trying to feed Quincy for days, and I was, you know, exhausted, and Kai wanted to help, and she didn't understand, like, having babies is really hard. And she, you know, she was just 22 months old. And so she saw a lot of like the hard of newborns with Quincy. I'm sure she doesn't remember much of any of it. And it was everything that she wanted to do to help. Like, you know, an older sister feeds the baby, and an older sister burps the baby, and an older sister holds the baby. Well, Quincy was just refluxing so badly and had such what looked like colic.
Right.
Kai couldn't get involved because she needed to be held in a certain way, and she needed a bottle in a certain way, and you had to remove the bottle immediately if she was about to throw up. And it was hard. And I think because of that, it was hard on my husband because he couldn't feed the baby. I had to do most of it. And Kai then became his responsibility. So he was with Kai and I was with Quincy. And then we really, like at about 34 days old, she actually wound up going to the hospital because she was refluxing so badly. She got something called laryngomalacia. And so basically the acid got on her larynx, her voice box, and made her voice box very weak. So she was having a very hard time breathing. And it was because it was swollen. And so we took her in, and they ultimately diagnosed her with laryngomalacia. So we were in the hospital for a week. So then Kai was not with me for a week, and my husband was with me, and then Kai was with our nanny or grandma and grandpa. And so that was hard. And so Quincy started getting a lot more attention. And we tried to make it easy for Kai and to, and he's here if you want him. We tried to make it easier for Kai in ways and spend time with her. But I do think that it probably did impact her, even though she'll never admit that. It had to have. And that was definitely hard. And as Quincy got diagnosed pretty early, she was around 6 months old. And Kai was the first person, I remember this so clearly, when we came home we got the diagnosis on the phone, and we were in a coffee shop. And when we came home, like, I fell to the floor. And I just remember Kai saying, we were both there, and our nanny was with Kai, and we were telling our nanny the diagnosis, and we didn't even know what it meant. And I just remember Kai saying something to us like, "Do you want Band-Aids for your eyes?" Because I was crying. I don't remember if he was crying or not, but I remember her saying that at 20, like at this point, she's probably like just about 2.5. And I remember her saying that, and like, how smart of her to be like, "You have a boo-boo," because she never saw us like that. And she ultimately just became a special needs sister. That was it. And it happened earlier than that because Quincy had so many challenges and delays, but it was very clear at that moment that Kai had a different responsibility, for sure.
So Quincy got diagnosed at 6 months.
Yep. I guess her official diagnosis was like 7.5 months, but like we got the genetic testing around 5 months, but they misdiagnosed her at first, and there was a whole thing about that. But like ultimately, yeah, around 6 months.
So the diagnostic journey wasn't actually that long for you.
It wasn't.
So 6 months, so this was in 2016 roughly?
2014.
14.
Yeah, so Quincy was born in 2014 March, and we officially got the diagnosis in November.
Okay, so genetic diagnosing was very new.
Very new. And it was actually, I did the NIPT, the non-invasive prenatal test, which Angelman is on it. But Angelman got put on it in 2014. And I was pregnant with Quincy in 2013. So it missed it. So I had that test. And because I had that test, our OB didn't feel we needed to do an amnio because that test was so good. And so we did an amnio with Kai, but we did not do one with Quincy, and it would have been picked up on the amnio. So we did not pick it up in utero. And then after that, when she was developmentally delayed, we saw a neurologist, and he said there's nothing wrong with her. Maybe she has mild cerebral palsy. And I was like, cerebral palsy? Like that's not something to take lightly.
Yeah.
And so ultimately, we really had to push for genetic testing. And it was mostly my husband that pushed. I was 100% in denial. The doctor said there's probably nothing wrong with her. And I took that and said there's probably nothing wrong with her. I feel like usually it's the mom that pushes and it's the dad that's in denial. It was not the case for us. He was very much like, there's something wrong. And I was like, there's nothing wrong. The doctor said there's nothing wrong. But as time, that was from like 3 months old. By the time we got to 5 months and we were getting the genetic testing, I knew there was something wrong. And so he pushed for it. And when we got it, the test came back normal because it was a karyotype and a microarray and not whole genome or whole exome.
Right.
And the karyotype was normal. The microarray was abnormal, but they actually only read the first piece of paper that came through on fax. That's back when there were fax machines. And the first paper said, X46XX normal. The second page said microarray, microdeletion, chromosome 15, rule out Angelman Syndrome and Prader-Willi. Because he never read that, he called us and said everything was normal. And then we said, can we see a geneticist? We really think something was wrong. Maybe we need whole exome. And he didn't recommend whole exome because it wasn't something that was easy to do. And so we saw a geneticist. The night before the appointment with the geneticist, he called us. We were in a coffee shop. And he said, a new test came in. And I asked to see it. It came in on the same day as the 2-week ago test. And it looks like she has a microdeletion of chromosome 15. She either has Angelman syndrome or Prader-Willi syndrome. And I Googled, which I should never do, Angelman syndrome. And she was like quintessentially on the list of everything that's Angelman syndrome. So we knew it wasn't Prader-Willi. We knew it was Angelman based on her symptoms. And we show up at the geneticist's office the next day, and he hands us a pile of papers from gene reviews on Prader-Willi syndrome. And I was like, you can take this back. She has Angelman syndrome. And he's like, well, we didn't do the methylation test. We don't know. I was like, look at her.
Yeah.
She has Angelman syndrome. She's blonde, blue-eyed, smiling and giggling as a 7-month-old baby. Pretty sure you know what she has. And so ultimately, that was what we got. And then we got the methylation. We were right. It was Angelman.
Seems like a standard of practice that for rare disease kids, they have that experience of going to neurologists usually, and the neurologist saying, they're fine.
Yeah. And I think that's changing now, finally.
Yeah.
I think because at the time, remember this was 2014.
Yeah.
Spinraza wasn't approved.
Yeah.
Luxturna wasn't approved. There was no approved gene therapy. Yeah. There was no approved ASO for a neurologic disorder. There was no enzyme replacement therapy outside of like Pompe for a neurologic disorder that's non-degenerative. It was not something that neurologists thought would ever be something you can treat. I mean, do you know how many parents are told, don't even do genetic testing. It helps science. It doesn't help your child.
People are,
5 years ago, people said that. And now neurologists, there's seizure screens, CP screens, autism screens, Simon Searchlight. Now parents can get a cheek swab and get their own genetic testing.
Yeah.
They don't need the referral to a geneticist or a neurologist. Where at that time, it was, why would you ever want genetic testing? She is not deformed. Yeah. She doesn't have a deformity.
Yeah.
She's anatomically normal. There's nothing wrong with her.
Yeah.
Maybe a little cerebral palsy, maybe a little autism. And you're just like, are you serious?
It'd be interesting. I wonder if people who have been diagnosed with cerebral palsy, how many of them have been misdiagnosed.
Yeah.
Because I just had an interview with, I mentioned Casey Walsh yesterday. Her daughter with SPG47 also initially diagnosed cerebral palsy. And how many, there must be like thousands of people diagnosed with cerebral palsy that have never had a genetic test.
Yeah. And part of our mission is to find them. And many of them are going to be in adult living at this point. Yeah. And going into those facilities and getting consent to genetic tests, these people is very hard, but it needs to be done. Because I also think just intellectual disability, autism, and cerebral palsy. Yeah. They say now 20% of all autism diagnoses are genetic. I would bet you it's 60, 70%.
Yeah.
Or more. It may all be genetic or epigenetic.
Yeah. So one of the things I follow is the whole genetic newborn sequencing with the icons and so forth. And living through it, things never move fast enough. But being objective about it, you can see the rate of which things are moving is super fast. And we're just kind of living through, the process. And I know the process is going to get to a point where when the therapies are available for N of 1, there is nothing that doesn't have an actionable treatment per se. Right? And right now, a lot of these screening programs, they only look for things that have an actionable treatment. So, I mean, one of the latest things that was added was SMA because of Spinraza and Zolgensma. But Phelan-McDermott syndrome, even though they're in a phase 1-2 trial coming to the end of it, is still not on the panel that they look for. And I know that's going to change, but the idea of actionable, looking for things that are actionable, I hope the definition of actionable just pretty much gets wiped off the table. Because there could be a whole generation of kids that say, well, we're not going to do, or we're going to do a genetic test. We didn't find anything. And that's the only genetic test that they're ever going to have.
Right.
Right.
Right. And also like having standards where you have to repeat them every 3 years because new genes get added to it every year.
Yeah.
And so what was negative last year is positive this year.
Exactly.
So it's really important. I also think that part of the issue with newborn screening is not only actionable, approved drugs. So you cannot get on the newborn screen if you don't have an approved therapy for the disease.
Yeah.
So in Angelman, it's a seizure disorder, right? So if kids go on a ketogenic diet, they can prevent ever having a seizure. So if they can go on a keto formula from birth, they can potentially never have a seizure. That's actionable. But it is not, ketogenic diet is not approved for Angelman syndrome, right? It's a known dietary modification for epilepsy.
Wow. So is Angelman on the panels that they look for in the new genetic newborn sequencing, like Guardian?
Yes, but only they have different levels. Level 1 for Guardian, so like opting in, when you opt in, parents have to opt into different levels. Level 1 is only for diseases in which there is an approved therapy.
Okay.
2 is diseases where there is approved therapies in development.
Okay.
And then 3 is not yet approved therapy, but, a community or something. What people don't understand is that having a diagnosis allows you access to be in a clinical trial for a potential transformative therapy earlier than you would necessarily have it. 2, having a community is therapeutic.
Absolutely.
That is an approved therapy. If you don't have a community of a disease in which your child lives with, and you don't know the best way to navigate all of the obstacles of rare disease, and every disease is different. Our kids do terribly with certain anti-seizure medications and do really well with others. Why should there be a trial and tribulation for our kids to have to suffer through drugs that don't work and actually hurt kids if they're part of a Facebook group that will tell you, never use this drug, Depakote, in your child with Angelman syndrome. It can be deadly.
When I asked this question of, I forget his name, Jeffrey, he was on a panel at Icons, and he is he's responsible for the public health for, I think it's North Carolina. And I had made that exact point to him, and he was suggesting that from a public policy perspective, it's a different perspective. From a public policy perspective, they have to kind of do what's right for everybody. And they have to balance all these different things, which, again, to me, is kind of like that white space that you're talking about.
That's living with the lowest common denominator.
Yeah.
That is not the way the world is. The world does not live with the lowest common denominator, right? The world should live with the average or above average and push everybody to do better. Yeah. So for me, if you take 100 families and say, would you like to be part of a community or would you like to not be part of a community? What percentage of them don't want to have a community in which they can ask questions and have support? 99.9% will say yes. Yeah. So what policy is that? Like, to me, that is the 1% that doesn't have Facebook or Instagram or TikTok or some access to the internet to be part of a community. Yeah. We're going to let 99% of the world suffer and not have access to support because 1% can't. Instead, why don't we do better by the 1% and give them access to those things, right? Like, and we're part of the Early Check, which is in North Carolina as well. We funded a project for a newborn screening with the Early Check system, which is what they're trying to do in the state of North Carolina. So we're on that. And, in order to be able to be at that next step of newborn screening approvals for a validated assay for newborn screening, which we have now done. But ours can never be on the newborn screen until we have an approved drug, and we don't have an approved drug. It's a vicious cycle that I think is actually backwards.
Yeah, this is where I see the bureaucracy and people who have been in it for so long and just don't know any other way.
And they don't live with rare disease. They don't understand. If you told me I could have a patient support group from day one when Quincy was diagnosed, when she started refluxing, and then I would have known how to handle everything thereafter for feeding therapy, because no one would give us feeding therapy because she was just a happy spitter. No, she's happy because she has Angelman syndrome and she doesn't know any better than to laugh. And she's in pain, but can't show us that because she's laughing. Yeah. That's not a happy baby. Yeah. That is a very severely painful baby that can't eat and has failure to thrive. Like, It's just insane.
Whenever I come up against things like that, I always tell myself, things are changing fast. It's like thinking back in history and everything that's happened for us to be where we're at today. And we're better off today than we were 100 years ago. And all the trials and tribulations that everyone went through, this is just history. That's how history unfolds. It's messy, and we're just living through that. But
it's like logic should just prevail.
That would be nice. That would be nice. We're getting there. We're getting there.
Patience is not my virtue.
So let's move over to, you've got Quincy's diagnosis, and she's 6 months old, and you go out and you Google and you start diving into what is Angelman's. From there, what's the next big step? Is that, was FAST just started at that time?
No, so FAST was around well before her diagnosis. I had nothing to do with starting FAST. It was
started. Paula?
Yeah. So it was started by a group of parents, 8 parents that were just amazing. And they were a paper came out showing that you can actually rescue the symptoms in a mouse model of Angelman syndrome with a gene therapy in 2011.
Oh, wow.
But the work had been done like 2007, 2008. And so a group of parents who were supporting, there was one organization in the US called the Angelman Syndrome Foundation. And this group of parents, 2007 and before, were supporting this organization and of course doing what they could to help Angelman. And parents just started kind of gathering of like reading papers and understanding the models. And the gene was only discovered in the late 90s. So like some like-minded parents that really had an interest in science started chatting and ultimately decided to start their own organization because the support organization was not very supportive of focusing on research or using the word cure or using the word transformative or, this is my understanding. I wasn't around at the time. And so there was a divergence at the time and a second organization started in 2008. And that was started by eight parents. And they all had different skill sets, a lawyer, an accountant, a real estate agent, they all had different skill sets in which they brought to the table, and they launched FAST. And then they started funding only research. It was not a support organization. It was only to support research and to support groups that were willing to think outside of the box. So I didn't know anything about FAST actually, because they didn't have the greatest presence on the internet. Honestly, at the time, like when I did a Google search, FAST didn't come up. Other organizations came up and unfortunately Wikipedia came up, which is the first thing that I read. But so the more I started talking to people in other organizations and realizing that they were nowhere near treating this condition. Like there was no knowledge of how to do that. I started then going to PubMed and just pulling everything I could on PubMed and calling the senior authors on the papers to say, okay, we have all of these animal models. We have all of this knowledge of the biology. You've shown you can treat it with an ASO, with a gene therapy, with a zinc finger. Why is this not in humans? So on my own, before I ever joined any organization, because I was so uninterested in being part of an organization, to be honest, I had so much on my plate. I was like in the height of my career. I was incredibly busy with Quincy, her sister, and my job that I loved, that I was not even thinking about what I was going to do with this. I just wanted to understand when we can put her in a clinical trial, because who wouldn't have a clinical trial for this? You can fix it. It's elegant. You could unsilence it. You could replace it. There's 10 different ways you could do this. The technology's there. Why is it not available for her? I was mind blown that this has been published now for 7 years and nobody has brought this to humans? Like, what is going on? So I just started calling everybody like, I must be mistaken.
Must be missing something.
Yeah, like there must be a trial somewhere. So called all of these researchers, and what was so clear was that there was this valley of death that we were sitting in which early research and discovery scientists, neuroscientists, and people that were really interested in understanding the genetics of this disease have done a ton of work to understand the biology, create the models, understand what drugs could and couldn't work. But they were not drug developers. They were in an academic lab. They got promoted by NIH funding. NIH money was far more green than foundation money. And they were just getting foundation money to get their next R01. They wanted their next big grant from the NIH because that would get them promoted. This is the academic life. And I get it because I was there for over a decade in academics. I was at Penn for a very long time, and I understand that. And that is where the world lives. And then the other side of the world was these doctors that were seeing Quincy who were like, we're waiting for these therapies because one day it's going to happen. But they weren't drug developers. They didn't know how to develop a therapy, but they can administer the therapy. And that's the valley of death. Where does neuroscience meet clinical medicine?
Mm-hmm.
And that's what most people think is a pharmaceutical company. And that's the savior that's going to take something out of an academic lab and give it to the neurologist.
Mm-hmm.
But guess what? Those people don't exist. They're not interested in Angelman syndrome. Angelman syndrome is 1 in 15,000 people. That's not going to be blockbuster. What company is going to actually put millions and millions of dollars into that, hundreds of millions of dollars into that, to develop a therapy that we have no idea if it's ever going to work?
Yeah.
No one. And so this is where we were in 2014. No one was interested in Angelman syndrome. There was not a single pharmaceutical company working on Angelman syndrome. And so this is when I realized, wow, we're in trouble. How are we going to do this? Who is going to do this? But we can't just wait for somebody to be interested. And so that's when I wound up joining FAST. I said, well, this isn't what I had my mind on, but if I can do anything to help this community, I make medical devices. I put them into patients. I can help patients. And my goal is to help kids. Maybe I can play a role here. Maybe I can help create a bridge of the valley of death, get pharma interested in doing this, become the low-hanging fruit, because SMA, there's industry-sponsored clinical trials. Why isn't there an Angelman? What can we do to make Angelman like SMA or like cystic fibrosis? We're not heard of. They are. Why? So I just jumped in. Ultimately, I wound up, getting very friendly with a lot of the great researchers that were doing a lot of work in Angelman, trying to figure out what they needed to get there, what was missing, what we could do to support them to get over that hump of mouse candidate to human candidate, and where should I go to do that? And I realized I'd have the most impact if I did that at FAST. So I wound up joining their board in 2016 and wound up becoming their chief science officer about 6 months later, and really was tasked with creating this roadmap. How do we get these 6 therapeutic candidates that have been working in animal models, or an idea that might work in an animal model, from mouse candidate to human candidate? How much is that going to cost? What is it going to take? And who's the best person to do it? And then how do we develop all the tools we need in order to become the low-hanging fruit for industry? Making sure the natural history study is appropriate, making sure that we have the right tools and models, the right biomarkers, the right endpoints, the right clinical trial design. What are we going to do on a clinical side to get pharma interested?
So when you joined the board, when you became the chief science officer, did you leave veterinary?
No. So I did this, my clinic day generally starts at 8:30 or so, 8 o'clock. So I would be a vet from 8 to 5 or 8 to 6. Uh-huh. I would come in early from 7 to 8 and do fast work. And then after I put the kids to bed around 7:30 or 8, I would start again and then work till like 11. And then Saturdays and Sundays, I would do the CSO work. So I was completely volunteer, unpaid, full-time veterinarian for 10 years. Yeah, so I didn't take any money at all. I was on the board as a volunteer CSO and just basically tried to figure out how to get this done and what it's going to cost and how we're going to get there. And so we created a path, we created a roadmap of all the different steps, made a budget, raised the money. Thank goodness we had an incredible donor who had a child with Angelman, whose parents happened to be part of a wealthy liqueur called Grand Marnier.
Grand Marnier.
Yeah. And really? We didn't know that, so because their last name isn't Grand or Marnier. So I just became really good friends with this dad, and we were very like-minded. He's French from the UK, and I was in New York, and him and his wife came to New York. They had a son with Angelman, and they just wanted to meet for lunch, and I met with them, and I showed them our roadmap on the back of a napkin with colored pencils to be like, this is what we're doing, this is what we're creating. I think that this is going to work. We need a plan, we need a path. We have never had one. Great work had been done, but we were at the point now where we needed a plan to move it beyond the academic setting.
Yeah.
How are we going to do that? And this is what we're thinking. And he's like, what is it going to cost? And I was like, it's going to be about $5.8 million, 24 months to get to clinical candidate for 6 platforms. And his family wrote us a check. I didn't know. He's like, well, I think we could do that. I was like, you? Who are you? I had no idea who this guy was.
As he's drinking a stifter.
No idea.
That's amazing.
Yeah.
So how long did it take you to get from getting that funding to putting it into action?
We had that conversation, I would say probably in May or June. We sat in front of his family foundation in July. And we went out to dinner with his parents that night after we presented in front of the family foundation with all of our scientists. They all came and they diligenced it and presented what their plan was that evening. And basically at dinner, they announced that they were going to fund the project. So we got a check after some contracting and things, but I don't know how long that took. I wasn't involved with that. And then right away, we got started in the fall and we got to our first clinical candidate in 6 months. We gave ourselves 24 months for 6. We got to first clinical candidate with the ASO in 6 months, and that was GTX-102. And so we decided now we needed to raise the money to get this into IND-enabling studies and to get it to people, because that was the next step. And we couldn't ask them for more money, but we couldn't ask the community either because we needed $10 million just to get through the monkey studies and all of the studies it was going to take for FDA. And so we needed to raise about 5 to 10 million. And that's when we decided to start a company. And that's when Genetics Biotherapeutics was started.
GeneTX.
Yes.
So when you started GeneTX, was that in partnership? Like you went out and you tried to find a partner to kind of be able to do that?
So that's when we decided to look at different options. So we ultimately wound up just doing an angel investor round, a Series A with angel investors, which was friends and family.
Yep.
The foundation put in a majority of the funds. So the foundation took the license to the ASO technology out of Texas A&M University, put that into, we call it genetics, GeneTX, genetics.
Right.
But yes, GeneTX is how it looks.
Right.
So we put that into genetics, and we basically started this for-profit company. And so between the collaboration of FAST, Texas A&M University, and a handful of friends and family angel investors, we had enough money to get through IND-enabling studies. So we raised that money together, got through IND-enabling studies from declaration of human candidate to filing our IND in about 17 months. And then we needed to raise the money for the clinical trial. That's when we decided to go out to bigger investors and say, how are we going to pay this? We wound up talking to all sorts of different types of investors, from small to large, including pharmaceutical companies. And there was a lot of interest. This is when we only had animal data. We had monkey data to show that it looked very safe, and it looked like it likely was going to be potentially very effective because the monkey has the same silencing mechanism. So we could show you can knock it down. We could show that it was well tolerated in the animals, and we could show that it lasted a nice period of time. And so there was real prospect for benefit. And so a lot of people were interested. This is 2019. So this is really where there was still a lot of interest in rare disease drug development before the crash. And so we had to make a choice. Do we want to bring in investors and take this on ourselves, or do we want to partner with a pharmaceutical company? One of the investors actually was a big investor in Ultragenics. I knew Emil Kakis at Ultragenics because I had talked to him 2 years prior when we were launching this campaign of getting to human candidate, and there was good data in the mouse. And he said, come back to me when you have human data. So I just remembered, like in the shower one morning, wait, maybe I should call Emil. I wonder if monkey data is close enough to human data, if he'd be interested in this. And so it was 2 years later, and it was before we had even started the process with the monkeys. And I decided, now that we have monkey data, maybe he changed his mind. So I gave it a shot, and I literally just cold called him. And I got on the phone with him. He asked me to talk to his business development team. My colleague Jennifer Panagoulias and I, who now still works with me at AS2 Bio, who's amazing. You should actually interview her. She's amazing. We got on a phone with the business development team at Ultragenics and went through the data that we had, got under CDA, and they said, are you available to come to Novato, California and present this in person? So 2 weeks later, we get on a plane, we go to Novato with we had a very tiny genetics team, which was 4 of us now. And the 4 of us went to California. We presented it all to Ultragenics, and then they gave us an option agreement to pay for the clinical trial with the option to acquire if they liked the phase 1/2 data. And they would give us the access to 18 full-time employees to support the program, which we only had 4 people. So we felt that would be amazing, because then otherwise we'd have to staff this whole thing up on our own. If we can use the CRO, the contract research organization, with the Ultragenics team that's very capable, we can make the decisions until we have clinical data in which they can opt in. Maybe that's the best path for us. So even though some people made stronger offers, we felt that was the right path. And other pharmaceutical companies showed interest as well. They just didn't seem as well fit. So we wound up partnering it with Ultragenics.
So everything was pre-negotiated in terms of what, if they were interested, like, was the purchase price kind of pre-negotiated, or was that negotiated after the fact, like after when you got to that point?
Yeah, so it was pre-negotiated, and I wasn't part of those discussions, but that's where business people come in, and I try to stay away. But yeah, so essentially there was an option agreement that they can opt in before we filed the IND, or they can opt in anytime after the IND. And if they did it earlier, it was cheaper, and if they did it later, it was more expensive. And they did it later. So they basically wanted to wait for human data.
Yeah.
And so they took the second option, and then of course more negotiations happened along the way, but they took it even before the end of the phase 1/2. They wound up taking it earlier than they anticipated they were going to take it, but not as early as they could have taken it.
Right. Yeah. So GTX 102 goes into human trials. It's an ASO. Is it kind of heavy loaded initially in terms of the injections, and then kind of you slowly spread it out?
Yeah, exactly. It's very much like what they do in Spinraza. So it's monthly dosing for 3 months, and then every 3-month dosing.
Okay.
That's the way the phase 1/2 started. It's been modified now. In the phase 3, they're doing monthly dosing, I believe, for 3 doses, and then they escalate every other month till they get to the top dose.
Okay.
And then they go to every 3 months. So it's a little slower of an incline. But the phase 1/2 was 4 doses over 3 months, and then every 3 months.
Did part of the trial cover stopping treatment and seeing kind of what happens in terms of, does efficacy come down? Is there a regression? Or?
Yeah, so that wasn't the intention. The intention was, once the kids were on treatment, ethically, if they were seeing benefit, to stay on treatment in a long-term extension study every 3 months. But unfortunately, there was a serious adverse event that happened in the first 5 kids that were treated, which ultimately put the program on hold until they figured it out. And that resulted in them having to stop treatment. So the first 5 kids, some of them got 4 doses, some of them 3 doses, some of them 5 doses, some of them 1 dose. But they had an adverse event, which was basically an inflammatory reaction, assumed inflammatory reaction to the drug, which ASOs cause inflammation. And it's concentrated at the lower spine because you inject it with an LP at the lower spine. And so the concentration of the drug sits there for a bit as it circulates to the CSF, but the highest concentration, it's diffusion, so it's a gradient. It's all in the lumbar spine, and then it moves its way up to the brain. So you essentially get more than 10 times the dose in the lumbar spine than you do in the brain. And so that high concentration at high doses ultimately resulted in irritation to the nerve roots and some inflammation, and they got weakness of their legs, the kids. And they put the trial on hold. Since then, that has happened now in multiple ASO trials. But this was the first trial that was publicly disclosed that this happened. We've now since learned, and now other public disclosures have happened. This is something that people are seeing.
And how do they
high doses.
And how do they get around it? Did they just kind of slow down the dosing?
Lower the dose.
Lower the dose and kind of spread it out.
And
over a long
so what Ultragenics did was they lowered the dose, and they follow it with a flush to kind of dilute it and push it higher, faster, and they tilt the kids in Trendelenburg position after dosing, so it goes up to the brain. Yeah.
And that?
And that wound up getting rid of the side effect. But because of that, the kids wound up coming off drug.
And
we saw them wane. So all the gains they had over those first 3-4 months went away. And so it was a forced on, off, right?
And they come back on.
And then they went back on about 2 years later. and they gained back everything they lost. So it was a very clear on-off on.
If there wasn't that negative reaction initially, would we have ever been able to get that information?
No. I mean, still the benefit would have been seen in the endpoints, but that like really was so profound that people were able to see that. And I think that gave Ultragenyx more confidence that this works. We just need to figure out a way around this side effect because this drug really works.
And what is it, developing an ASO cost-wise, manufacturing, and so forth?
Now, ASOs aren't terribly expensive compared to a gene therapy.
Right.
So, they cost, depending on how much you're making, for a phase 1 clinical trial in the administration is actually more expensive than the drug itself compared to a gene therapy, which is much more expensive.
Is it just because they're Angelman kids and you can't do it otherwise?
Yeah, you can't tell them to sit still with a needle in their back.
Gotcha.
Yeah.
Okay.
And some kids on the ASO are now able to do it without anesthesia because they're learning that they can sit still and actually get it done.
So everybody who's been part of the phase 1-2, has the phase 3 started yet?
Yes, it's done.
It's done.
It's done. So we'll have the data in Q4.
What is the process like once the data is published? Let's say best case scenario, everything's published. Is the next step going to the FDA?
Yeah. So then they'll have to disclose their data, and then they'll go to the FDA and file NDA, which is a new drug application.
And then it takes
about a year.
A year.
Yeah. So I think it depends on for them, because we have rare pediatric designation, fast track designation, and breakthrough designation, it might be shorter.
Okay. And in that interim, are they allowed to?
They keep dosing the kids in the trial.
Yeah.
But they can't dose new kids.
They can't add any kids to the trial.
New kids. Once it's approved, then everybody will have access. But that's still a process of once it's approved, then insurance companies have to start. So that's another process.
So that's a negotiation process.
That's happening in that year.
Okay. So Ultragenyx is going to be negotiating with insurance companies to try to find as much as insurance companies will pay for it.
I believe that's the process. I don't really know the process, but I believe it happens in parallel. So that the hope is when it's approved, it's a really smooth launch for everyone who wants it to have access to it in the jurisdiction in which it's approved.
So now genetics is kind of on its own. It's running on its own. It's running kind of outside of FAST. It's being pushed by Ultragenyx. The money that came in from Ultragenyx to FAST, now FAST has a war chest, call it. I know it goes fast, but now FAST has now started AS2 Bio and is now funding research for other methodologies.
Exactly. So we started an accelerator to take those 6 therapeutic platforms I told you to get to human candidate. Yeah. The ASO was first. The AAV gene therapy was second.
Okay.
A CRISPR was third.
Yeah.
And a hematopoietic stem cell gene therapy was fourth.
Okay.
So we took the other 3 programs, since the ASO is now with Ultragenyx, and did the same thing we did with genetics and created 3 new companies.
Okay, one for each.
One for each, under the umbrella of the management portfolio company, AS2 Bio. AS2 Bio is essentially the holding management company, and they have 3 portfolio companies under it. Mavericks, which holds the AAV, Courageous, which holds the CRISPR, and Transformatics, which holds the HSC program. And I'm the Chief Development Officer of AS2 Bio, responsible for all of the non-clinical studies and clinical.
Okay.
So I basically am responsible for the strategy and the execution of all of the IND enabling studies and how that's going to support the clinical program, and then the strategy of clinical trial design and how we're going to get these programs into the clinic. So basically there's 3 more genetics companies.
So are you essentially using the same playbook?
Yes, exactly. With different modalities, different platforms, so different manufacturers, but similar clinical trial design, similar engagement with the FDA.
What stage are all 3 kind of running in parallel?
So the AAV gene therapy is in clinical trials right now. So we've dosed patients. So that program is in phase 1-2.
Okay.
And then the CRISPR is in IND enabling in monkeys. So that we hope to file the IND mid-2027, so about a year from now.
Okay.
And then the hematopoietic stem cell program is behind that.
The gene therapy, AAV9?
It's like AAV9. It's called Q68, which is a variant of AAV9, but it's basically the same.
Lumbar puncture as well?
No. This is delivered by ICM delivery.
Oh, into the base.
Yeah, because it's for the brain, not the spinal cord.
Right.
Yeah.
Which is what I think SPG50 is.
SPG50 is doing lumbar puncture.
Oh, is it?
Yeah, that's all the way down here.
Right. But there's a sac over here or something that you kind of go into.
And that, or you can go right through the
Which is
skull,
ICV.
ICV, right?
Exactly.
So ICM and ICV have the same distribution.
But ASOs, they won't deliver that way?
I wish, because you have to do it every 3 months. It's not safe. So they need to put a port in to do that.
Gotcha.
Which I think should be the next step ultimately, is adding a port.
Okay. And that's kind of permanent and...
Yeah, it can go like right under the scalp, right here, right into the ICV. And then all you need to do is do an injection right into it every 3 months, and no anesthesia, nothing. And it's
then,
under the skin.
And then the biodistribution will be...
Just to the brain.
For the CRISPR therapy. Oh, that... Is it CRISPR the gene therapy? No, the gene therapy, and then you got the base. Is it base or prime, or is it?
It's actually neither. It's a Cas9 knockdown because we have the unsilencing mechanism. We don't have to change a single nucleotide. So it's actually a knockdown approach.
Okay. Is that considered easier?
Much easier.
Yeah.
Much easier because it can help all the patients. We don't have to be individualized based on your mutation. Yeah, so much easier. And so a single treatment gene therapy for some people is going to be easier, less burdensome, and potentially better than an ASO. And for others, the reversibility of an ASO and the fact that you can give it or not give it and change your mind is something that might be better for a family. For Angelman, we're the same size as SMA, a little bigger actually. So potentially it's a $5 billion market. It's a profitable disease. If you have a disease of 10 or 20 or 30, then it's hard to justify profitability.
Right.
When you have a disease of thousands, it's much easier to justify profitability. The problem is where people slow down is in getting it to profitability.
Are the failure rates changing with the introduction, like with AI now being part of the process? Are they finding a higher pass-through rate?
I don't think that there's data to support that yet. I think that genetic therapies have a much lower failure rate, period. And that's because you know your target. When you're just targeting drugs for Alzheimer's disease, when you really don't know your target and you don't know what causes it, so you can't justify how to stop it, that's much harder. The failure rate there is very, very high. But if you have a genetic cause of Alzheimer's that you determine, that you can target preventing it from ever resulting in Alzheimer's, the success rate of that would be dramatically higher. So I think the statistics are like 1 in 15,000 drugs are ever approved that are developed.
Oh, wow.
For regular disease. Yeah. I think for rare disease, it's probably closer to, 1 in 50 to 1 in 25. It's much, much more likely as you get into clinical trials that a drug will be successful in a rare disease, especially if there's no approved therapies in that disease. The bar is very low in terms of, for hypertension, you have to beat the other hypertension drugs for it to be approved.
Yeah.
In Angelman, you have to beat nothing because nothing is approved. So if you can show an improvement in communication, an improvement in activities of daily living, an improvement in self-feeding, an improvement in social interaction, in aberrant behaviors, that's approvable. So rare disease drugs are much more likely to be approved because the impact they will have on everyday life when there's no approvals is tremendous.
The playbook that you guys have developed and now you're kind of sharing, in what form is that? Is there going to be like an AI bot that's available for new foundation parents that will just take them through the process? And
I wish that would be lovely. Right now, there's one bot that's not AI. No, that doesn't exist yet. And, we try to do everything we can to share through, obviously multiple conversations. I talk to a new rare disease group every week,
right?
More than that, probably. Guiding them on their drug development plans, their roadmap. How do they create their own roadmap? What do they need to do? Where do they start? So it's always from like, where do I start? My kid was just diagnosed to how do I get a drug into my kid? And that's a huge process. But there are these rare disease boot camps that exist now, multiple of them through the rare, the Epilepsy Network, and then through Ultragenics, through Mazi Therapeutics, through Global Genes. So there's a lot of these groups that are helping advance other groups. And a lot of that started with, what did FAST do? How did FAST get there? How do we build on that? So we go and we speak at all of those conferences and those meetings. We give, lots of interviews and lectures and talks about where to start, what you need. There's not exactly a playbook per se, like on a piece of paper, as much as a lot of lectures and talks and diagrams and images of kind of what our roadmap looks like, and then people are creating a roadmap that looks very similar. But a lot of it is really just piecemeal. And we do need to create something that's a lot easier to just plug and play, but who has the time to do that? We're trying to fix Angelman syndrome.
I want to thank Dr. Allyson Berent for this conversation, and more importantly, for everything she has done for the Angelman syndrome community and the broader rare disease world. Her work with FAST, AS2 Bio, and the families pushing science forward is a powerful reminder of what can happen when urgency, science, and parent advocacy come together. These conversations matter because they help families understand what is possible, where the science is going, and why this moment in rare disease therapy is so important. If you found this interview meaningful, please subscribe to Generation Hope on YouTube, like and share this video, and help us reach more families, researchers, clinicians, and advocates. You can also visit generationhope.co to watch more interviews, sign up for the newsletter, and follow our weekly updates on the latest in gene therapy, RNA therapy, newborn screening, clinical trials, and rare disease research. Thank you for watching, and thank you for supporting Generation Hope. And remember, stay hopeful.